rated as excellent. But the
Featherization, we think that featherization, what the possibilities are for featherization in BrainGain and how we are trying to do it in Artinis Medical Systems. I first will give you a short introduction because we're not so famous as Philips. About what we're doing. We're a young small company and we're located close to Nijmegen. We're a spin-off company of the University of Nijmegen, and therefore we have two groups of products which are both medical but maybe a bit strange combination. The first one is the golden standard X-ray phantoms. They're used for quality control in radiology and mammography. You can use them for testing, by example, contrast detail properties of radiology systems, for homogeneity of the detectors and a lot of other tests. But we're not, because of this group of products, into BrainGain, but for near-infrared spectroscopy. We have different types of systems. For this, one is left, that's the classical system we have. It's a big system, and we have a small one in which we are unique. And I will tell you a bit more about those and about the technique. Well, what is near-infrared spectroscopy? It's an optical, non-invasive method. You can place a sensor and a sensor and a light-emitting probe on the skin. The light goes into the skin, travels around, scatters, and is absorbed, and is detected by the system. And the absorption is dependent to the properties of the tissue, and especially to the oxygenated and the deoxygenated hemoglobin. Both have a different absorption spectrum, and therefore we can discriminate the concentration changes of these. And you can use it to measure the oxygenation and hemodynamics in living tissue. Well, how can you use this? First, you can also use this on muscles during sport exercises, in rehabilitation, but also on the brain. You can measure the activation of the cortex. If you do, by example, finger tapping with your hand like this, you can measure at the left cortex, if you do it at the right hand, the activation. Left you see the signal. The red line is the oxygenated hemoglobin concentration change, and the blue line is the deoxygenated hemoglobin change. And at every L, the vertical lines L, you see an activation where the subject started with finger typing. What you see is that the blood level goes up, and that you see especially in the oxygenated hemoglobin concentration. Compared to EEG, what most people know, this is a bit slower signal. The response time to the max is about 5 seconds, but it has a very good spatial resolution, and that's why it has other possibilities than EEG. Well, what do we do in BrainGain? First of all, we contribute our knowledge about NIRS and our measurement methods for people who would like to know more about it. And second, and that is the main part, is the development and the validation of new NIRS BCI methods. One of the questions we got from the BrainGain board is what do we do and what do we expect from our BrainGain partners? Well, it was quite hard to answer it, but we thought about it, and we think that just one group is using NIRS in BrainGain for one specific application, but they're doing a nice job, and that's the Sanmartí clinic. Also, TNO had a NIRS system, but they don't have people anymore to do it, I understood. So what we would like is that more groups would have or will use NIRS in BCI, and we're talking about it with some groups, and maybe it will give something. Well, what's our role in BrainGain? How do we do featherization? Well, what I mentioned already is that we contribute our knowledge about NIRS. Other deliverables or things we did is the software coupling via field trip. This is nice because we can use it to do other things. Also what we try to do is make a hardware coupling with the system of TMSI. And this is also for the same purpose, but yeah, hardware coupling might be nicer than via software. We have a study about the advantage of using NIRS and EEG together, and a study with measurements of imaginary movements. That's yeah, to look if you can help impaired people with using NIRS. Another thing is that we're trying to improve the brain signal measurement, and we're trying to develop a portable brain measurement method. And last two things I will tell a bit more about. Well, what you do if you measure NIRS, you measure at the skull, and you measure through the skull, and also the fluids in between the cortex and the skull. And in this fluid there's also a little bit of absorption. This will have influence to the signal you measure, and therefore we tried to make a correction for this. And how we do that is to have a second channel, which is much closer to the receiver. And since the depth of the place where you measure is dependent to the distance between the receiver and the transmitter, we can measure here also only before the cortex, so you have only the surface signal. And how do this look in practice? If you look to the signals, it's like this. This is a raw signal with the classic method with one receiver, one transmitter. So we're measuring cortex and surface signal. This is the shorter distance, so this is what we call the reference. And well, it's quite simple. If you subtract those both signals, you can see that you get a better signal than in the above two graphs. And what we normally do is we average, and then you get a very clear signal, and this improves the NIRS measurement a lot, especially if you want to do BCI, because you don't want to have false positives. Well, we made a nice application for it. You can play a game with it. We call it Brain Snake. Unfortunately, we can't show it in a demo because you need a lot of concentration for it, but it's quite nice. Other applications at the end users are now that it's used for neurofeedback and in the study for the imaginary movement measurements. The other thing we're doing is the development of the portable brain measurement method. Why are we doing that? Well, the both systems we have are not appropriate to do this. The big system, you need fibers, it's heavy and handy, it costs a lot of time to set up. The other system, it's made only for muscles. You cannot put it onto the head because the skull is bended. It's much easier in real life because it doesn't hurt, and it's a small sensor. So, and you can walk around with it. It's connected via Bluetooth. And it has the same applications as I told before. That's BCI and neurofeedback. How do we do that? That's our portable, as I showed on one of the first slides. That is a sensor at the size of a cell phone, and in that one we have the sensors internal in the portable itself. But now we make one with external sensor. We'll show it in a demo where you can do to it. But it's still a prototype, so we have to tackle some problems, like the integration of the reference channel. And if you put it on your head, you have hair in between the sensor and the skin, so you can only measure now on the front at the forehead. And if you want to do other measurements, yeah, that's still not possible, unfortunately. Well, what do we see as opportunities in BrainGain? for the current BrainGain projects, we think that are a lot of nice ideas, but it's quite complicated. It's not, they're not easy to set up at the users. It costs a lot of time to build up all the experiments, and especially if you want to do it in home situations, you have to, all the systems are big. So we think it should be simplified, and also it's, well, mainly working on ideal subjects. There are quite a lot of subjects which are non-responders, at least for NEARS, and that has to be improved, that we can measure more people. And we think for Artinis Medical Systems there are also quite a lot of opportunities, because we got a lot of knowledge, and we can bring this to the researchers and our end users. And it gives us also more sales, which is very nice, of course. It gives awareness at the customers and the end users, and we bring this knowledge to other projects like Focus Food and Cognition, I3B, and Quantivision, and other projects. Well, that was my presentation. I hope it was a bit clear.